Imagine meeting your exact duplicate. Same face, same voice, same memories. Sounds like a sci-fi movie, right? But with cloning, it could be reality. Welcome to Explain Me This, where we unravel the mysteries of the universe. Picture this. The year is 1996. The world is introduced to Dolly, a cute, fluffy sheep that shook the scientific community to its core. Why, you ask? Because Dolly wasn't just any ordinary sheep. She was the first mammal cloned from an adult cell. Suddenly, the realm of science fiction seemed within reach. The possibility of human cloning was now on the table, and it sparked a global debate that continues to this day. But here we are nearly 3 decades later, and we still haven't cloned a human. Now, you must be wondering, why is that? Well, it turns out cloning isn't as straightforward as it seems. It's a complex process fraught with technical barriers and ethical dilemmas. The process of reproductive cloning involves inserting DNA from a mature somatic cell into an egg cell. Sounds simple enough, right? But when it comes to humans, we haven't quite cracked the code yet. The challenges are many, and they are significant. We're talking about risks of loss of life, violation of human dignity, and concerns about creating individuals with unique thoughts and opinions. And then there's the question of necessity. Do we even need to clone humans? With alternatives like induced pluripotent stem cells, human cloning has become less of a necessity for research purposes. Plus, human germline genome editing has been gaining more attention recently, raising its own set of ethical and safety concerns. So as you can see, the curious case of human cloning is far from being solved. It's a fascinating topic, filled with intrigue, ethical debates, and scientific challenges. Before we dive deeper, remember, if you're enjoying this video, smash that like button, leave a comment below, and subscribe for more mind-bending content. And hey, if we ever clone humans, maybe your clone will do it too. So how does cloning work anyway? Are we talking about a mad scientist in a lab coat, or is it more complicated? Indeed, the science behind cloning is quite fascinating, and no, it doesn't involve a mad scientist, well, not exactly. Let's simplify it. The process of cloning begins with a mature cell, which contains all the genetic information needed to create an organism. This cell is also known as a somatic cell, and it could be anything from a skin cell to a liver cell. The DNA from this cell is then extracted and inserted into an egg cell. But here's the twist, the egg cell has had its original DNA removed, so the only DNA in this egg cell is from the somatic cell. Now this egg cell starts behaving as if it's been fertilized, and it begins to divide, eventually forming an embryo. If all goes well, this embryo can then be implanted into a surrogate mother and carried to term. The result is an organism that is an exact genetic replica of the original donor of the somatic cell. Sounds simple, right? Well, not quite. Cloning is a highly intricate process, and there are numerous factors that can go wrong. For one, the success rate is relatively low. Remember Dolly the sheep? She was the first mammal cloned from an adult cell back in 1996, but it took 277 attempts to get it right. Furthermore, when it comes to humans, the process becomes significantly more complex. Our bodies are made up of trillions of cells, each containing a unique combination of genetic information. This makes the task of successfully cloning a human being a monumental challenge, to say the least. So, yeah, cloning is a bit more complicated than just copying and pasting DNA, but hey, nothing worth doing is ever easy, right? Now that we've got the science down, let's talk about why we haven't cloned a human yet. Spoiler alert. It's not because we're afraid of creating an army of zombie clones. Instead, there are 2 major roadblocks that we've bumped into on the road to human cloning: ethical dilemmas and technological hurdles. Now these aren't your everyday problems. They're the kind of issues that keep scientists awake at night, pondering the implications of their work. Let's start with the ethical dilemmas. Human cloning opens a can of worms that many people are uncomfortable with. For one, there's the risk of loss of life. Cloning techniques aren't foolproof, and failed attempts could result in loss of potential human life. Then there's the issue of human dignity. There's something unsettling about creating a carbon copy of a person. It's like reducing the complexity and uniqueness of human life down to a set of instructions that can be replicated in a lab. Would a clone have the same rights as a naturally born human? Would they have unique thoughts and opinions? Or would they just be a copy of their genetic donor? These are questions that don't have easy answers. Next up, the technological hurdles. Despite advances in cloning technology, we're still a long way off from being able to clone a human. The process of transferring DNA from a mature somatic cell into an egg cell is fraught with difficulties and has yet to be successfully achieved in humans. There are also concerns about the health and lifespan of clones. Remember Dolly the sheep? the first mammal cloned from an adult cell, she lived only half as long as the average sheep and suffered from several health issues. It's possible that similar problems could occur in human clones, making the process not just unethical but also unsafe. So, as you can see, cloning is a bit of a Pandora's box. Open it and you might unleash a whole lot of trouble. But don't lose hope. Science has a way of finding alternatives when a door closes. And in this case, the alternatives are pretty exciting. When we hit a wall with human cloning, we didn't just pack up and go home. Instead, we looked for other intriguing possibilities. And one of these is the concept of induced pluripotent stem cells. Now, these aren't just your average cells. These are special cells that can be coaxed into behaving like embryonic stem cells. And why is this cool, you ask? Well, these cells have the potential to develop into any cell type in the body. Yes, you heard right, any cell type. This means they can potentially replace damaged cells in the body, opening doors to treatments for a host of diseases. So, while we may not be creating clone armies, we're certainly making strides in medical breakthroughs. But the story doesn't end there. Lately, there's been quite a buzz around something called human germline genome editing. In simpler terms, it's about editing the DNA of an embryo to bring about desired genetic traits. Sounds like science fiction, doesn't it? But it's very much a reality. However, as with anything this groundbreaking, it comes with its own set of ethical and safety concerns. Should we be playing God and altering the course of human evolution? What if there are unforeseen consequences down the line? These are questions we need to grapple with as we plunge headfirst into these uncharted territories. We need to strike a balance between pushing the boundaries of scientific discovery and maintaining respect for the sanctity of life. So, while we might not be cloning humans anytime soon, the future of genetic research is still bright. And who knows, maybe one day we'll find a way to meet our clones after all. So there you have it, folks, the why, the how, and the not yet of human cloning. It's a wild ride, isn't it? Indeed, we've navigated the intricate labyrinth of human cloning. From the science that brought us Dolly the sheep, to the ethical dilemmas and technological hurdles that stand between us and the potential of creating a human clone. The journey's been nothing short of a roller coaster ride, filled with fascinating twists and turns. In this whirlwind journey, we've discovered that cloning is more than just a sci-fi concept. It's a scientific reality, yet one that is fraught with complexities. From the intricacies of transferring DNA from a mature somatic cell into an egg cell, to the monumental task of replicating the uniqueness of human life, cloning is a Pandora's box of scientific challenges. We've also delved into the ethical labyrinth that surrounds human cloning. Questions about the sanctity of life, the violation of human dignity, and the unsettling notion of creating individuals with unique thoughts and opinions have emerged. These are weighty issues, requiring thoughtful and balanced discourse. Yet amidst these challenges, we've also glimpsed the future. Alternatives like induced pluripotent stem cells have opened up new pathways, making human cloning unnecessary for research purposes. Human germline genome editing, while raising its own ethical and safety concerns, has also stepped into the spotlight. So, as we wrap up, let's remember that the world of science is ever evolving. Today's impossibility might be tomorrow's breakthrough. The question of human cloning remains open-ended, a testament to the endless possibilities and challenges that lie in the realm of scientific exploration. Remember, science is all about asking questions and pushing boundaries, so keep questioning, keep exploring, and keep tuning into Explain Me This for more mind-blowing content. Catch you in the next one.